Hi, welcome to Goal 2 or Unit 2, Biochemistry. This includes atomic structure, bonding, and molecules. So we're going to talk about the basic atomic structure, what an isotope is, what an ion is. We're going to talk about how the electrons and work in bonding different things together, including ionic and covalent and hydrogen bonds. We're going to talk about pH, organic molecules, building blocks, and finally the structure and function of enzymes. This is quite a long PowerPoint, and don't be afraid to listen to just part 1 and then take a break before you do part 2. So, what is matter? Matter is anything which has mass or weight and occupies space. Matter is all the living things we can see, feel, and smell. That doesn't have to be living. A desk, a banana, an apple, a pen. It's all the stuff around us. In order to understand the properties and reactions of matter, it must be divided into simpler, less complex substances. So it's easiest to work with these definitions if we start with the basics. So the development of atomic theory starts with this. All matter is made up of elements. Elements are the simplest form of matter which cannot be changed by further chemical or physical methods. You can't break an element down into further parts and still be the element itself. An atom is the simplest form of an element. Elements are the basic units of all substances, and the most common elements in humans are oxygen, carbon, hydrogen, and nitrogen. Those 4 elements make up 95% of humans. So atoms are the smallest units of matter. Each atom contains protons, which have a positive charge, neutrons with no charge, and electrons with a negative charge. The structure of the atom will be described shortly, but elements are made up of the same kind of atoms. So that carbon atom makes up carbon, for example. You can't get any smaller than that. Usually, atoms do not exist all by themselves, but are hooked up with other atoms. Molecules are defined as any 2 atoms hooked together. It could be 2 of the same or 2 different ones. Okay? They include things like fats or carbohydrates or oxygen, O2 for example. Compounds and molecules are sometimes used interchangeably, but compounds, the only difference is that a molecule can be 2 of the same atoms and a compound has to be 2 different atoms. So O2 can be a compound or a molecule, but CH4 can only be a compound because there's 2 different atoms. When you write this down on a piece of paper, the atom is the capital letter, and the small number next to it is the number of atoms in that molecule. If there's nothing written there, then there's just 1. So in this molecule, C6H12O6 has 6 carbons, 12 hydrogens, and 6 oxygens. H2O has 2 hydrogens and just 1 oxygen. If the letter is 2 letters, like Na, where the first letter, the N, is capitalized and the next letter is not, that's 1 element. It just has 2 letters in its atomic symbol. CH4 has 1 carbon and 4 hydrogens. NaCl, it has 1 sodium and 1 chloride atom. Alright, the parts of the atom again are the protons, neutrons, and electrons. In the nucleus of the atom, that's where the protons and neutrons hang out. Protons have a +1 charge and neutrons have no charge. So in this picture, the red pluses are indicating the protons and the yellow circles are indicating the neutrons. The electrons spin around or zoom around the nucleus in shells, sometimes called valence shells, sometimes called orbitals, but they zoom around in these shells basically with a certain number. The first shell around the nucleus can only hold up to 2 electrons. There can be none, 1, or 2, but as soon as you have 3 electrons, that third one's got to go to the next shell. The second shell can have up to 8. It can have 1, 2, 3, 4, 5, 6, 7, or 8, but it can't have more than 8. The third shell can have up to 18. However, once it hits 8, some start to fill the fourth shell, up to 8 I think, before the other 10 to make the 18, and the third one gets filled up. Because we're only going to talk about atoms usually in the first 3 rows of the periodic table, we're never going to have to worry about a fourth shell, or hardly ever. And basically the atoms are happiest when their outermost shell is full. So the first shell has up to 2, the second shell has up to 8, and then the third shell has up to 8. They're most balanced or stable when those outer shell is full. That's where the 2-8-8 rule comes in. That's what makes atoms the happiest, when their outer shell is full. So we remember those 3 numbers, 2, 8, 8 for the first, second, and third shell. You'll be good to go. Alright, the atomic number of an atom is the number of protons. You cannot change the atomic number, or the number of protons, without changing the element. Number 7 is nitrogen. Number 6 is carbon. If you add a proton, you're not carbon, you're nitrogen. So the number of protons in an atom, or the atomic number, which is the same, the atomic number is the number of protons, does not change. Okay? Atomic number is the number of protons, and it does not change. In a neutral atom, the number of protons and electrons are equal. So if you know the atomic number, you know the number of protons and the number of electrons. It's the same number. The atomic weight of an atom, or its mass, is the weight of the protons, neutrons, and electrons all added up. Protons and neutrons each have an atomic mass of 1. Okay? Electrons have an atomic mass of 0.0000, all like 17 zeros and a 1. So electrons hardly weigh anything at all. And that's going to become important because you'll be able to figure out how many neutrons are in the average atom by simply subtracting the atomic number from the atomic weight. So here's a periodic table, and notice there's rows and columns. The first row has a hydrogen on the far left, number 1, and number 2 is way over on the far right, helium. Number 3 is in row number 2, lithium, Li, and then Be, beryllium. And then you kind of skip that middle space and you get to boron, carbon, nitrogen, oxygen, fluorine, and neon. Number 5, 6, 7, 8, 9, 10, etc. Okay, we are really mostly going to be concerned again with the first 3 rows, and then potassium and calcium, K and Ca, and we'll talk about those quite a bit, and iron, number 26. Most of the rest of them we're not going to find in the human body, and if you do, it can be very dangerous because they're toxic. So mostly we're not going to have to worry about very many of the bigger elements. So each little square in the periodic table, the top number is the atomic number. Remember, that's the number of protons. Also the number of electrons in a neutral atom. The next is the atomic symbol, which here is C, and what that stands for is the name, carbon. Okay, so C is for carbon, and most atomic tables will have the symbol and the name. The number on the bottom is the atomic weight. This is again, you add up the weight of all the electrons, neutrons, and protons, but electrons practically weigh nothing. So that number 12 there, if you round it to the nearest whole number, 12.01 would round to 12, and you subtract the number of protons, which is the atomic number, 6, you get the number of neutrons. So carbon has 6 protons, it also has 6 neutrons, because 12 minus 6 is 6. If we go back to that periodic table, you can find carbon up here, number 6. Notice the 6 on the top, the carbon, then the word carbon, then 12.01. If you look at fluorine, number 9, it has 9 protons and 9 electrons, and it weighs 19. 19 minus 9 is 10. It has 10 neutrons. So you can use the periodic table to figure out the number of protons, number of electrons, and number of neutrons. Okay. Alright, so the number of protons in this would be 6. In a neutral atom, there's also going to be 6 electrons. And to figure out the number of neutrons, you take the atomic weight, 12, round it to the nearest whole number, and subtract the atomic number, 6. So 12 minus 6 would be 6 neutrons. The other thing is, what does it look like? How does the electrons look around that nucleus? So in this case, there's 6 protons in the nucleus. There's going to be 2 electrons in the first shell, because they only can hold up to 2. And then because it's got 6 electrons, there's 4 left for that outer shell. It is not particularly stable or happy there, because remember the 2-8-8 rule. It would really like to have that outer shell full at 8, or get rid of it so the outermost layer shell of the first one, which is 2, is full. Alright, next thing we're going to define is the difference between an isotope and an ion. An isotope is basically an atom that's got more or different number of neutrons than average. We just figured out on the previous slide that carbon has 6 neutrons. Normal carbon has 6 neutrons, but every once in a while you come across a carbon that's got 7 neutrons or 8 neutrons. If it had 8 neutrons, it would weigh 14 instead of 12, because remember, neutrons and protons each have a weight of 1 atomic mass unit, and 8 plus 6 is 14. Well, atoms or isotopes with those extra neutrons, sometimes they lose them, and when a neutron flies off, it releases a little bit of radioactivity. Okay, so carbon-14, half of those isotopes are going to lose a neutron, or that carbon atom. About half the extra neutrons will be lost every 5,200 and some years. The half-life of the carbon isotope, what that refers to is how often a neutron leaves. So carbon-14, if you had a carbon-14 in your body 5,000 years from now, you'd have carbon-13. Another 5,000 years, you might be down to carbon-12 for that one carbon atom. So basically they know about how much carbon in the world, or making up everything, is carbon-14. So let's say I'm made up of 100 carbon atoms, and 2 of those are carbon-14, because 2% of all the carbon on Earth is carbon-14. And if I died today and you dug up my bones 5,000 years later, or roughly 5,000, I'd only have 1 carbon-14 atom left, because one neutron has left, and so one of them is down to carbon-13. So basically that's how they date bones. When they dig them up, they know that they started with 2% carbon-14, and they can use a little Geiger counter to measure the radioactivity left. They can determine, oh, well, half of his carbon-14 is gone, this must have been here 5,000 years. If another half is gone, he must have been here, or this body, this organism, whatever, must have been here 10,000 years ago. And it's pretty accurate. So this is how they date things, using the carbon-14 isotope thing. Isotopes are kind of cool because they can use them for all kinds of things. Because of the radioactivity, when a neutron leaves, you can catch this on film. And so they use radioactive labeling to test us medically. X-rays, radiation for cancer treatment, because radiation can mutate DNA. We use them for PET scans. We can test your thyroid by giving you radioactive iodine and then following where it goes. We can use gamma rays to sterilize equipment. There's all kinds of things they use isotopes for. You can actually get a degree in nuclear medicine and learn how to use isotopes in medicine. Ferris has a great program in that, so check it out if you're interested. And they teach you how to handle these things very safely, obviously, but they're very well paid jobs in nuclear medicine. All right, the other thing we're going to define here is an ion. An ion is an atom that has gained or lost an electron. Okay? Gained or lost an electron. Remember, the protons stay the same, whether they're isotopes or ions. Because you change protons, you change the type of atom. All right, so if you've gained an electron, you have an extra electron and you still have the same number of protons, you're going to be negatively charged. If you lose an electron, you'll have one more proton than electron, so you'll be positively charged. So ions are sometimes defined as charged atoms because they have a positive or negative charge. In this example right here, sodium has one electron in its outer shell, and it doesn't like that because that outer shell is not full. Chlorine has 7 in its outer shell, and it doesn't like that because it's not full. So if sodium gives one up and chlorine gets it, they're both happier because chlorine now has a full outer shell of 8, and sodium, its outermost shell now is full at 8 because it lost the one in the third shell. So everybody's happy, but sodium is going to have a positive charge and chlorine is going to have a negative charge because there's either a gain or a loss of electron, and they will stay together because of that positive and negative opposite attract thing. Okay, so ions are charged atoms. They have gained or lost an electron. And this is basically what's going to become an ionic bond, which we're going to talk about here in a few minutes. In us, ions are not called ions, but electrolytes. So floating around in your blood, the sodium ion is referred to as the sodium electrolyte. Basically, when we eat salt, and salt gets into water, it dissolves or disassociates into its ions. And we need some. We need sodium, chloride, potassium, magnesium. We need all kinds of ions to make sure everything works. Okay, you can't send a nerve impulse without sodium and potassium ions. You can't clot blood without calcium ions. All these ions are needed for the body. And they're pretty easy to get. All you got to do is eat. When you sweat or you lose a lot of liquid, you don't just lose liquid, you also lose some of these ions, which is why they tell you to eat Gatorade or Pedialyte or whatever to help restore your electrolytes. So we call them salts, but in water, they disassociate. So besides conducting nerve impulses and muscle contraction, we need the ion iron to carry oxygen. We need all of them together to help maintain osmolarity, which we're going to talk about in goal 3. Okay, and a whole bunch of other things in the human body require these electrolytes. You can take them as supplements as well, but most of us, if you're eating a normal, healthy diet, you should have no trouble getting these. We will talk further about electrolytes in lab. So at this point, you should be able to define the following items shown on this slide: matter, element, atoms, molecules, and compounds. Use the periodic table to determine protons, electrons, and neutrons, and define ions and isotopes. So this is a good stopping point if you'd like to pause after part 1. The next section is all about bonding. Alright, bonding is basically moving around the electrons. Remember, protons are going to stay the same because that keeps the element whatever element it is. And neutrons aren't involved in the bonding process at all. They don't leave the nucleus. So electrons in the outermost shell is really what determines whether an atom bonds ionically or covalently. Okay? Remember that 2-8-8 rule. Atoms like to have their outermost shell full at either 2 if it's the first shell, 8 in the second shell or 8 in the third shell. The types of bonds we're going to talk about is ionic and covalent, and then we're going to talk about hydrogen bonds, the weakest one. So let's start with ionic. Some atoms in their outermost shell have just 1 or 2, or they have 6 or 7, so they need 1 or 2 to make that outermost shell full. And usually these two types of atoms get together. Okay, so in our example from before, and you can see it here, sodium has 1 electron in its outer shell. 1 happens to jump over to chlorine, who has 7, means that the outer shell of sodium would have 8, and the outer shell of chlorine would have 8. Both of them lots happier. There's a net positive charge now on the sodium, and a net negative charge on the chlorine, and they hover together because opposites attract. They're both called ions, okay, because again, an ion is any atom with a charge. But an ionic bond forms because one atom is positive and one is negative. Okay, so here's another picture of ionic bonds. One atom donates an electron, the other atom receives 1 or 2. The opposites attract because of the opposite charges. And most importantly, if you put that ionically bonded molecule in water, they come apart into its ions. Okay, they come apart or dissociate into the sodium ion, which is written here as Na+, because it's got a positive charge, and chloride negative because it's got a negative charge. That's going to become important when we talk about pH. All ionic molecules dissociate in water. They come apart into the ions. Covalent bonds, it's usually between atoms that have 3, 4, or 5 in the outer shell. Too many to lose or too many to gain compared to the ionic bonding molecules. So instead of giving up or getting that many, they share. So ionic bonds are donating and receiving electrons. In covalent bonds, they're sharing. So the atoms actually get closer together and the bond is much stronger. When you put a covalently bonded molecule in water, they do not dissociate. It's not coming apart because these are sharing electrons. So hydrogen in this picture has 1 electron in its outer shell. It doesn't like that. It would be happier to either have none or have 2, fulfilling the 2A8 rule. So what it does is the atoms get very close together, and these blue little dots, the electrons, will zoom around like a figure 8. They'll both be going around both nuclei of the 2 atoms. So if you count the number of electrons in the outer shell of the one on the right, it's 2. If you count the number of electrons in the one on the left, it's 2. So again, they're very happy at that 2-8-8 rule, okay, but they're really just sharing those electrons. Okay, again, this bond is much stronger because of that sharing. So again, they share electrons. They do not associate in water and are quite strong. This picture right here on the first one is CH4. Carbon has 4 electrons in its outermost shell, which are colored red here. Hydrogen each have 1 electron in its outer shell. So what happens is the hydrogens share their 1 electron with the carbon, and there's 4 of them. So if you count the number of little dots around the carbon, red, blue, blue, red, blue, red, red, blue, there's 8, okay? And if you count the number of dots around each hydrogen, there's 1 red and 1 blue, or 2. So they both fulfill that 2-8-8 rule, and everybody's happy. In water, H2O, the figure on the right, the outermost shell of oxygen has 6 electrons. 1, 2, 3, 4, 5, 6 little blue dots. Hydrogen, the atom in the hydrogen is depicted as an X in this one, has just 1. So it shares with oxygen, 2 hydrogen atoms share a pair of electrons with the oxygen. So if you count the number around oxygen, it's 8 now, and if you count the number of electrons around hydrogen, it's 2. So everybody's happy. Hydrogen's got 2, oxygen's got 8, both fulfill the 2-8-8 rule. This is water, and you can't break water, right? Water is water. You have to have a heck of a lot of energy to separate the oxygen and hydrogens out, away from each other in a molecule of water. Okay, so again, covalent bonds share pairs of electrons. In ionic bonds, electrons are donated or received. There are a couple of special cases of covalent bonds where instead of one pair of electrons being shared, you share 2 pair or even 3. So you can have double covalent or triple covalent bonds, which are even stronger than single covalent bonds. Okay, very strong. This is carbon dioxide. Carbon shares 2 pairs of electrons with each of the oxygens. And again, very hard to break that up, which is why it's building up in our atmosphere, because not very much breaks this down. Alright, so that's covalent and ionic. The last type of bond we're going to talk about is hydrogen bonds. Hydrogen bonds occur between 2 different molecules or within a really large one. And it's basically a very weak attraction between hydrogen and oxygen, or hydrogen and nitrogen. So in this little picture, the red with the 2 little white balls, the little red ball with the 2 little white balls, each one of those is a molecule of water. And the hydrogen bond is the very weak attraction of the hydrogen from one molecule of water to the oxygen on the other part of the water. So if you look at this where the hydrogen bond is labeled, the hydrogen of the top water molecule is slightly attracted to the oxygen of the second water molecule. Very weak, but it's there. And one of the ways you can easily demonstrate this, if you take your hand and you put water in it, you turn your hand over, almost all the water just falls right out. But there's that one little drip that's kind of hanging onto your skin. Okay? Even though gravity wants to pull it down, and if you flick your hand, you can get some of it to go away. There's a little bit of water that will stay up there. That's hydrogen bonds. There's a slight chemical attraction, or an electron attraction, between the water molecule that you put in your hand and a water molecule on your, or the skin molecule itself, the skin cell itself. A little bit of steam or a change in pH, you're going to break this hydrogen bond easily. Okay, the water molecules, you get them a little bit hot, they're going to become, instead of liquid, vapor, right? So hydrogen bonds are pretty weak, but they allow for things like tiny insects to walk on water, or surface tension, that's called surface tension of water, or even the adhesion. When I told you, my example, when you turn your hand over, you'll still get one drip hanging on your hand. It's what allows newts and other salamanders to walk on your ceiling. Okay, it's the weak attraction between water molecules in the ceiling itself and the pads of their little feet. Okay, so hydrogen bonds are powerful even though they're very, very weak. And we'll talk about them more because one of the things they do is, in this picture on the right here, is they fold up proteins and DNA. And if proteins or DNA didn't fold up into particular shapes because of these hydrogen bonds, they wouldn't work. If our DNA didn't work, we'd be dead. If our proteins didn't work, they'd be dead. We'd be dead. So even though weak, hydrogen bonds play a very important role in our lives. Alright, so again, you should now be able to tell me which type of bond is strongest. And that is covalent, right? It takes energy to break them, and when you do break them, you usually get a really little bit of energy. So, for example, food is covalently bonded because it's got energy in it. We call it calories, but in our body, breaking those covalent bonds is what gives us the energy. Which type of bond dissociates into ions in water? That's the ionic one. Okay, salt dissociates into the sodium ion and the chloride ion. Which type of bond shares electron pairs? That would be covalent. Which type is the weakest and an increase in temperature will cause it to break? That's the hydrogen bonds. Okay, they are the weakest, but very important for life as we know it. Okay, another little review here. What happens when ionic compounds are placed in water? They dissociate. Those dissociated atoms in solution are called ions or electrolytes. Here's a little picture. The ionically bonded molecule sodium chloride, you put it in water, will dissociate into the electrolytes sodium and chloride. HCl is also an ionically bonded molecule. When it splits apart in water, it becomes the hydrogen ion and the chloride ion. So it's ionic just like salt. However, the presence of the hydrogen ion makes the solution acidic. And that is going to be our next topic, pH. We'll also be talking about this in lab, so don't forget to use your lab book or check out the lab and do it if you're an online student or in class for the face-to-face students. All right, so what is pH? pH is basically the measurement of how many hydrogen ions are in a solution. And they do this using the logarithmic scale. And we're not going to talk about logarithms because that's horrible. Basically, you need to remember that the more hydrogen ion is in a solution, the pH goes down and it becomes more acidic. So it's kind of weird. More hydrogen ion makes it more acidic, and the pH goes down below 7. 7 is considered neutral, and above 7 is considered alkaline or basic. Pure distilled water is supposed to have a pH of 7, be neutral. We like acidic stuff. We call it sour, but we like the flavor of tomato juice and grape juice and orange juice and that sort of thing. We don't eat a lot of basic stuff. Milk could sometimes be a slightly basic, but soap is basic, and nobody likes their mouth washed out with soap, right? Tastes nasty. Bleach is very, very basic. And we don't want to eat too much really acidic stuff or basic stuff. That would kill us, because all of the molecules in our body that use hydrogen bonds to fold up would break. Hydrogen bonds break if you change pH. And you remember from Unit 1 that our pH is supposed to be 7.4. And even a little variation beyond that is quite deadly to us. Okay, so how do you determine if something is an acid, base, or salt? Acids are when the hydrogen ion is present. Bases are when you see the OH ion, which is called the hydroxyl ion. When that's present, the solution is going to be basic and the pH will go up. If neither one are present, it's just a salt, so sodium and chloride, neither one of those is going to change pH. So the change in pH when you add salt, 0, zip, nada, nothing. Okay? You add something that will disassociate and release a hydrogen ion, it's going to be acidic. The hydroxyl ion is going to be basic. Neither one, no change in pH, you just call it a salt. So you can kind of go through the flow chart here, look at a molecule. If it's ionic, go to number 2. If it's not ionic, it will not dissociate and will not change pH. So covalently bonded molecules should not change the pH of a solution. After dissociation, is there hydrogen ion present? If yes, it's an acid. If no, go to number 3. Is there an OH, a hydroxyl ion present? OH negative, yes, it's there. You have a base. If not, go to number 4, it's a salt. Okay, so you can look at these molecules and you should be able to determine if they are acids, bases, or salts, or if they're not, if they're covalent, they're not any of those. Okay, so the next slide will show you the answers to these. So don't look at the next slide if you want to practice these. or stop it here, see if you can figure them out, and then click to the next slide. So here's the answers. KOH, for example, potassium plus the hydroxyl ion is what happens when it's put in water. That OH makes it a base. OH, basically, if a molecule has carbon, C, oxygen, O, or N, nitrogen, CON, it's almost always covalent. The hard part is hydrogen can go either way. So H2SO4, the SO4 is covalent, it won't come apart, but the hydrogen will come off of it. Okay? If it's got OH, that OH won't break up because it's covalent, but it will break away from whatever it's bound to, because that is the hydroxyl ion. Okay, this is another good stopping point. Part 2 is going to talk about macromolecules and building blocks, so if you need to take a break, this is a good time to stop it before you go on to part 3. So the last part, or the second part of the biochemistry section, is the building blocks of macromolecules. There are basically 4 macromolecules, and each macromolecule is made up of building blocks, and we're going to talk about those. So you're going to hear about these over and over and over, so just put them in your head and keep them there. Building blocks are monomers that when you connect them together form a polymer. So think of like a bead making up a necklace. The bead is a monomer, and when you hook them together you get a polymer, or the whole necklace. Okay. The 4 building blocks of macromolecules in AMP include sugars, which when you hook them together make carbohydrates or polysaccharides, fatty acids, which when you hook them together make lipids or fats, amino acids, which when you hook them together make proteins, and nucleotides, which when you hook them together make nucleic acids. All 4 of these are covalent. All require energy to make, and when you break them down, release energy. So when you break those bonds, you release energy. So like this picture says, the food we eat are covalently bonded molecules because when we break the bonds in them, we get energy. And that's why food gives us energy. In general, we eat the macromolecules, but our cells eat the building blocks. We can eat a potato, but it's too big to get into our cells. We got to break all that carbohydrate down into individual molecules of glucose. So we eat the potatoes, our cells eat the glucose. All building blocks are organic. Now, organic means a lot of different things to a lot of different people, but in this class, organic means there's a carbon and a hydrogen in that molecule. If both are there, it's organic. If they are not both there, it's inorganic. So water, oxygen, carbon dioxide, and salts are all inorganic because water doesn't have any carbon, oxygen doesn't have carbon or hydrogen, carbon dioxide doesn't have any hydrogen, and salts don't have any carbon or hydrogen. So it's not that organic and inorganic are more or less important. We need both of them, right? We need water, we need oxygen, we need salts. We also need organic molecules, carbs, fats, and proteins to eat. Okay. So let's talk about the first one. The building block is glucose. The macromolecule is carbohydrate. Glucose is the most common monosaccharide. There's also galactose and fructose. And they make up all these good foods that we love to eat. Plants put together glucoses in the form of starch or cellulose. Okay? We can take starch and break them down into individual molecules of glucose. We love starch. Cellulose, even though it's glucose molecules hooked together, notice they're in the opposite orientation, we can't break it down. We call it fiber. It's in our diets, and that's good because it helps keep us regular and keeps basically poop from getting too hard. Okay, but we can't break down cellulose. The third type of macromolecule carbohydrate is glycogen. This is how we store glucose. So if I eat extra glucose, I can convert it to fat, but I also could hook them together in a molecule called glycogen. This is the storage form of glucose by animals. Okay, plants store it as starch and cellulose. Animals store it as glycogen. Okay. We only store enough glycogen for about 12 to 24 hours because it takes a lot of water to store it, which would make us weigh a lot if we had to store all our extra energy as glycogen instead of fat. Fatty acids are the building block that make up lipids. And fatty acids, here's a picture of one. It's made up of carbon, hydrogen, and oxygen. Not very much oxygen compared to carbohydrates. And this is a long-chain saturated fatty acid. Here's an unsaturated fatty acid. The difference between the two being this one's got a double bond. Remember, double covalent bonds versus single covalent bonds? And who cares about the double and single bonds or whatever, but saturated fats tend to be less healthy for us than unsaturated fats. So all the diet people out there, the USDA and all the doctors, go home and eat unsaturated fats instead of saturated. Can you guess which one tastes better? Saturated tends to be solid at room temperature, like the fat on meat or chocolate, Crisco, and butter. That stuff tastes really good. Unsaturated fats tend to be more liquidy, and they include things like olive oil and fish oil. Fish oil is actually polyunsaturated fatty acids. It has 2 or more double bonds, and that's even better for you than monounsaturated. But people tend to like chocolate a lot more than fish. Anyway, when you hook these fatty acids together, you get triglycerides, okay, which is the macromolecule. Okay, so we store fatty acids in the form of triglycerides, and we eat them in the form of triglycerides, break them down to the fatty acids so our cells can use them. Number 3, amino acids, the building block, make up proteins. This is the structure of amino acids, and you do not have to memorize it, but notice that unlike carbs and fats, there's a nitrogen in amino acids. So this is the only building block so far that has had nitrogen, and that's what makes amino acids different from fatty acids and carbohydrate or monosaccharides like glucose, that nitrogen. And to get rid of that nitrogen, if we have to clip it off or break them down, that's why we produce urine, because urine is mostly urea, which is the form of nitrogen that is removed from our bodies as waste. Anyway, you hook a bunch of amino acids together, you get proteins. Proteins look like this beaded necklace until hydrogen bonds fold them up into beta-pleta sheets or alpha helices, or anyway, proteins do not work, do not do functions unless they're folded up properly. So it shows an active functional protein versus a denatured one. Denatured means all the hydrogen bonds are broken. And you can do that 2 ways: heat up the protein or change the pH, which is why your body temperature doesn't want to go above 98.6 very much, or you don't want to change your pH beyond 7.4. Keeping those 2 things at homeostasis keeps our proteins working and keeps us working. Last, DNA and RNA are made up of nucleotides, A's, G's, C's, and T's, and we'll talk about this more when we get to goal 3, but nucleotides make up nucleic acids, and this is a picture of DNA on the right. But DNA and RNA, both important molecules for passing on genetic information and gene expression, are made up of the building blocks nucleotides. So in summary, the building blocks, glucose, fatty acids, amino acids, and nucleotides, make up the macromolecules, carbs, fats, proteins, and nucleic acids, respectively, and they do stuff for us. All of these things can be eaten for energy. However, mostly that's what we use sugar and fat for. We tend to use the proteins that we eat for functional things like hormones or enzymes or transporters or building muscle before we use them for an energy source. So carbs and fats, those covalent bonds in those molecules, are broken to give us energy. We do use some fat to make cell membranes and steroid hormones. We use amino acids in the form of proteins to make hormones and enzymes, receptors to do stuff. And we use the nucleotides to make RNA and DNA, so for cell replication and gene expression and that type of thing. These two tend to have jobs more than the carbs and fats, where their main job is to, let's face it, they taste good. That's a good energy source, right? You have to have some food so you can run around and build snow people. Alright, the last thing we're going to talk about before we call this good is, what does an enzyme do? Enzymes speed up the rate of reactions. Okay? You can't possibly imagine how long it would take to convert cookie dough, into cookies if you just left them at room temperature, right? We put them in ovens because all of a sudden that cookie dough in 10 minutes or less turns into a delicious cookie. And that's kind of what enzymes do. They convert cookie dough into cookies in a time that makes it, useful. You can eat those cookies 10 minutes later, right? If we had to wait until that potato broke down into a pile of glucose for our cells to eat, it would take possibly years. So enzymes speed up reactions that otherwise might occur but not fast enough to keep life as we know it existing. Enzymes are usually proteins, and they convert substrates into products. So the starting substance is the substrate. What you end up with is the product, without changing themselves. So when you put cookie dough into an oven and 10 minutes later you pull out cookies, the oven acts like an enzyme. It's still an oven. You can put in more cookie dough and get more cookies 10 minutes later. It did not change. Okay, now certainly you need some things to make sure your stove works. It needs to be plugged in or hooked up to the gas or whatever. But it itself didn't change. Okay, so the enzymes in your cells are very much like ovens. They speed up the reaction without changing themselves. Enzymes are also very specific to the substrate. Remember, the substrate is what they start with, like the cookie dough. So in this case, the red circle is not going to fit on the enzyme on the right. It's only going to fit on the enzyme on the left because they're shape specific. So it would be kind of like you having to have an oven to cook, one oven to cook chocolate chip cookies, one oven to cook molasses cookies, and one oven to cook Christmas cookies. Okay, because enzymes are specific to what they start with. And that specificity is a shape difference. Okay, they fold up to particular shapes. They bind their substrate, like the picture on the right there, and convert it into a product. So the green and red circles became a double purple shaped item there. Here's another picture. Okay, the enzyme binds the substrate, which is the little bluish gray thing, and it causes a reaction, and then it lets it go, the product. It releases it. It can do this over and over and over as long as the enzyme is working. Okay, so they're specific to a substrate based on their shape. What bonds give proteins their shape? Hopefully you remember this from just a few minutes ago. Hydrogen bonds. Okay. What kinds of things break those hydrogen bonds? A change in temperature or a change in pH. So this, like what we talked about in Unit 1, this is why it is so important that we try to maintain 98.6 and pH 7.4. If you think about it, if you're walking outside and then walk inside, you might have a temperature change of 50 degrees, right? Or even more. Your house is set at 70, and sometimes in the winter here it's 0, right? Or minus 10. That's an 80-degree difference. And yet your body temperature stays at 98.6 because your enzymes wouldn't work at 0 or at 70 or at 100. It needs to be 98.6. So we do all kinds of things to maintain body temperature. Another thing enzymes need to work sometimes are what's called cofactors or coenzymes. Basically, these are little molecules that bind the enzyme that make it work just a little bit better. The most common of cofactor, vitamins and minerals, okay, which of course are needed in our diet, and they're needed for enzymes to work. Okay, you don't eat vitamins and minerals to get calories. You eat them because it makes our enzymes work better. Okay, you don't need very much, but if you don't have them, some of our enzymes don't work as well as others. Enzymes are also controlled kind of by negative feedback, just like homeostasis is. Once enough product builds up of an enzyme reaction, it shuts off the enzyme itself. Okay, if you don't need any more product, then why keep making it, right? Your cells are 100% efficient, not like people, right? So as soon as enough of the end product has built up, it usually stops the first reaction in the pathway, so you don't waste energy, or the cell doesn't waste energy doing something it doesn't need. All right, that is the end of Unit 2. This is quite a long unit. There's lots of different resources out there. Check out some of the animations. Use your reading assignment and associated Quizlet. There is some other information on Canvas that might help you. Don't forget your lab book. There's a lot of good information about your lab, about these topics in your lab, and exercises that you'll do that might help you remember them a little bit better. And as always, you can always email me questions.